under video.
Open this. Hi, good afternoon, sir. And good afternoon, dear friends. And we will
that one, last one, big size. It's a
okay, good afternoon once again. We will continue with the topic that we discussed in the morning. So now the topic for the discussion is the characteristics of insect pollinating plants. See, this morning we have discussed some example for the plants, normally using the insect as a pollinating agents. So to make insect as a pollinating agent required some characteristics that only we are going to discuss here in detail. And once this question asked in the board examination, the characteristics of the insect pollinating plants. See, normally such a type of plant produce large flowers. That is to attract the insect only. It should be visible to the insects. And also very, very colorful. Then fragrance, it produces what? Very good fragrance, very rich smell, and rich in nectar, produce huge quantity of nectar as a reward for the pollinating agents. And night blooming flowers, normally they are white in color because white color is actually that contrasting background for this darkness, so that insect can be attracted. When flowers are small, as I told you, the number of flowers are more, and it is clustered in the form of what? The inflorescences. Then flowers pollinated by the beetle fly produce a very foul smell, because that beetle can be attracted only by these foul smells. That is why this that beetle pollinating flowers are normally producing such a very foul smell. Then provide nectar and pollen grain as a reward to the bees by the plants. Whenever these bees are helping the plants to do pollination, as a reward, in back, these plants are giving a huge quantity of nectar as well as the pollen grains. And pollen grains are sticky in nature. For what you can say, insect pollinating plant, they produce what? The pollen grains. Normally it is very sticky in nature, so that it can stick the body of this insect, and hairy and spiny body of the insect can stick the pollen grains in their body. You know that body, I told you in the morning, the surface of its body is with so many hairy material, so that the sticky pollen grain can stick on their body so easily. And honeybees are having a pollen bag on their legs. They are having pollen bag, that is to keep the pollen grains, particularly in the leg of the workers. Then this is what the body of see honeybee, and this is what the pollen bag. You can see the pollen bag on the leg of what this worker honeybee, and it is to keep the pollen grains, and for the what you can say the queens and drones. And if flowers are small, normally it appears in the form of clusters. And this is what the body of insect, you see it is a hairy one, so that the sticky pollen grain can easily stick on its body. And see, these are all pollen grains, and it is stick to the body of these insects only. So this is what the way these pollen grains are carried from one flower to another flower of the same species. And it is what the night blooming flowers are, that flower which use the beetle as a pollinating agent. Normally it produce what? Very foul smell to attract the beetles. And see the structure, the external features of the pollen grain, having so many hairy things on its periphery. So such type of what irregular surface only provide help them to stick on the body of such a hairy insects, right? And come to the what you can say, the plants, those who use the water as a pollinating agent, characteristics of water pollinating plants. And this floating type of male gamete only normally produced because it is using, as while discussing about the Wallisneria, I told you that male gamete producing flowers are always under the water. After producing this gamete, because of its lightweight, it will float on the surface, and this water wave or this water current only taking this pollen grain to the stigma region of another flower. The pollen grains are very sticky in nature, and pollen is long and ribbon shaped so that it can float on the surface of this water. Then pollen is protected with a mucilaginous cover. A mucal cover is there over what this pollen grain, and it is also for its protection only. And Wallisneria, then Zoosteria, that is what the seagrass, these are depending water only as a pollinating agent. Then come to the another one, the characteristics of the plant. Those who are using wind as a pollinating agent are anemophilic type of plants only, right? And for wind, reward is not at all required, so that never produce nectar. And color is also not there because no need to attract the pollinators for this pollination, because it is using what abiotic factor only as a pollinating agent, that is wind only. Then pollen grains are light. It is very essential to take through the wind. Then it is non-sticky pollen grains, never stick each other because it is carried out by the wind only. And pollen production in large quantity. Whenever wind is taking the pollen grain to the stigma region of another flower, naturally chances are there to lose a lot of pollen grain compared to the number of what you can say, the female reproductive gamete ovum, its number is too high. And single ovules present only in one ovary because chance of sticking the pollen grains to the stigma region is what very minimum. Then normally these are all inflorescent and spread stigma and style to trap the pollen grain. That the stigma region is feathery type so that it can easily trap the pollen grains while taking this pollen by the wind. And then come to other than pollen and nectar. Other than pollen and nectar, right? That means normally plants are providing pollen and nectar for a insect as a reward for the pollination. But other than this pollen and nectar, that bees are attracted to the flower, and they are what you can say, helping the plants by doing this pollination, right? And some plant provide a safe place to lay egg for insect as a reward. They are not giving, plants are not giving that pollen grain or the nectar. Instead, they are giving a very safe place for laying their eggs. Example, relationship between the largest flower, Amorphalus, and moth. See, this is what the Amorphalus, and there we can find the egg laying area. This is what the inside only. They are laying egg inside this moth. So the relationship between the this moth and this Amorphophallus plant is what very good example for such example. And yaka and moth, they cannot complete their life cycle without each other. They are helping each other to complete their life cycle, and such type of relationship is there between the yaka and moth, because moth select only the yaka plants to lay their eggs. Then moth deposited egg in the flower locule. Locule means what? The compartment where the ovule is located. And this moth deposited its egg in this locule of the flower, and in turn get pollinated by the moth. So flower is providing the space, locule, for its egg laying, whereas in return that moth is helping the plants to pollinate the flowers. And larvae of the moth comes out of the egg as a seed start developing. Whenever seed, after this pollination, that ovule only develop to become the seed, and whenever the seed get developed inside that ovary, that egg also has become the larvae, right? And this is what the Amorphophallus and moth relationship. It is not for the nectar or other thing. It is only for getting a suitable place to lay the eggs only. And this is what the relationship between the yaka and moth. And see, this moth is laying egg in the locule. Locule means the compartment where ovule is located. And this is what the insect as a pollinating agent, that is endomophily. Next topic is what the use of insect as a pollinating agent, that only we call it as what the endomophily. Entomology means what the study of insects, and endomophily, this is what the pollination, type of pollination using this biotic agent called insects. And bird as a pollinating agent, that is called as what? Ornithophily. Ornithology is what the study of bird. Ornithophily, where this is what pollination only, where that birds only used as a pollinating agent. Then here bat can also pollinate some of the flowers, and that is termed as what? Chiropterophily. Chiropterophily. Here is a pollination by using the agent called bats. See, some insects are not at all pollinated by the insects because they are pollen robbers or the nectar robbers. See, normally that plant is providing nectar and the pollen grain for the insect as a reward for the pollination. But some cases, some insects are taking this pollen or the nectar without doing this pollination. And that only we call it as what? The pollen robbers or the nectar robbers. And consume pollen and nectar, but never do pollination. This insect, you see the insect, they are collecting the nectar, not from here. They are making a small hole here and sucking directly nectar from the flower, that never involved in the process of this pollination. They are only, we call it as what? The nectar robbers or the pollen robbers. And some insects are used for the pollination without giving any kinds of rewards. An example is what? The sexual deficit. And here the flower is look like what you can say, female insects only in its marking, color, and all other structure. That petals of this flower exactly look like what? The female flower in its look, then color, marking, etc. So naturally, that male insect will be attracted and sit on it. While sitting, naturally, what? The process of pollination will happen. And the plants are making insect to do pollination without giving a reward. And example is what? The sexual deficit. Then come to the outbreeding devices. Outbreeding devices. Devices present in the plant supporting outbreeding. And if that plant is going for self-pollination for many generations, chances are there to lose a lot of vigor than their body. A lot of good character, that only we call it as what? The vigor. So many such vigor will go if that plant is going for inbreeding for many generations, and that only we call it as what? The inbreeding depression. Chances are there to lose many good vigor from the body due to the continuous inbreeding. So most of the plants are having some device. Particularly in the flower, it is having some device to avoid such type of inbreeding, and that only we call it as what? The outbreeding devices. So these are the devices to prevent inbreeding or self-pollination. And these are the devices present in a plant to discourage self-pollination, because self-breeding result inbreeding depression. It makes the plants to lose many useful characters. That only we call it as what? The inbreeding depression. Then pollen release and stigma receptibility are not synchronous. This is one methods to avoid inbreeding. Pollen release and stigma receptibility both are not getting matured at the same time and are not synchronized. Maturation of male and female gametes are in different time, so that no chance of any kinds of self-pollination. And anther and stigma plays in a different direction in a flower. In a flower, anther and stigma both are there and both are getting matured also in the same time, but anther is directed to one direction and the stigma to another direction. So here chance is very less for such type of self-pollination. And another one is self incompatibility. Incompatibility means what? The mismatching. Self incompatibility, it inhibits the pollen germination and pollen tube formation in the stigma of the same flowers. That whenever that pollen grain fall on the stigma of the same flower, because of the cell incompatibility, that pollen grain never germinate and never produce what? The pollen tube. And that only we call it as what? The self incompatibility. And next one, the production of unisexual flowers. Such a plant produce only the unisexual flowers, so that no chance of any kinds of self-pollination. Only one type of flower produced by the plant, either male flower or the female flower. And example, that only we call it as what? The dioic. Example is what? The maize and papaya. Papaya, male plant is there and female plant also there. Then male plant produce only the male flower and female plant produce only the female flower. It is as what you can say, outbreeding device only, avoid the self-pollination, right? Then come to the pollen pistil interaction. Now we are discussing about the pre-fertilization event, the events happening before the fertilization only. So first, the pre-fertilization event is formation of both pollen grains and the ovule, microsporogenesis and megasporogenesis. And second event is what? The pollination, transfer of pollen grain from a flower, the place of origin, to the stigma region. That only we discussed now. Now we are going to the next one, that is pollen pistil interaction. See, after this pollination, pollen grains will be there on the surface of the stigma. So there will be an interaction between the pollen grain and stigma region. And this interaction is very essential to identify that pollen grain is really from the same species or not. If it is from some other species, it is not permitted to sprout and go for the further development, because chromosome number will be different. You know, they are having different makeup, genetic makeup. It is not permitted to that sprout and go for the fertilization. That is why such type of interaction will be there between the pollen grain and pistil just after this pollination, and that only called as what? The pollen pistil interaction. And interaction always through some chemicals only. Some chemicals are there on the surface of that stigma region, that only helping to identify the pollen grain of the same species. And this interaction, we call it as what? The pollen pistil interaction. See, after pollination, there is a conversation between the pistil and pollen grains. That pollen grain, after this pollination, pistil will ask many things related to what you can say, the pollen grain, and chemical conversation only. And it is called as what? The pollen pistil interaction, exclusively to identify the compatible from the non-compatible. Means what? Matching one. What type of pollen grain will match with the stigma? The pollen grain of the same species only. So they want to identify whether this pollen grain from the same species or not, whether it is compatible or incompatible. For that only this interaction will be there between the stigma and this pollen grain. And the pistil has the ability to identify and recognize. Pistil only recognize and identify the what you can say, pollen grain of the same species. And interaction is due to the chemical present in the stigma surface only. And pollen grain also having what? The chemical material on its surface. The chemical present on the stigma is generally called as what? The pollen germinating fluid. There is a particular fluid on the surface of the stigma, that only making the pollen grain to sprout and go for what you can say, fertilization. And the chemical nature of this fluid is not same, varies species to species. This is what the way it identifies the pollen grain of the same species. Some chemicals are there in the stigma region, and this chemical only help to identify the pollen grain of the same species. And this pollen, why it's called as pollen germinating fluid, once it's identified this pollen is from the same species, it can make the pollen grain to go for germination. That is why we call it as what the pollen germinating fluid. And this is what the pollen grain attached to the stigma region. Stigma is what you can say very sticky in nature. So pollen grain can stick on its surface. Once it is attached to the stigma region, there is a type of conversation between the stigma and this pollen grain chemically, and identify the compatibility. Once it's identified, it's compatible, it permits the pollen grain to go for its sprouting and produce what the pollen tubes, right? Then come to next topic. Once it is identified as compatible, that stigma permit the pollen grains to germinate and produce what the pollen tube. This is also a pre-fertilization event solely. After pollination, after interaction between the stigma and pollen grain, that pollen grain start germinating, right? So if it is compatible, if it is matching both male gametes and the stigma region, pistil permit this pollen grain to germinate and produce what the pollen tube. And acceptance or the rejection of the pollen grain by the pistil is due to the nature of chemical present on the stigma. As I told you, if it belongs to the same species, naturally the stigma permit the pollen grain to sprout. Otherwise, never permit it disintegrate on the surface of the stigma itself. Because when this wind carry that pollen grain, chances are there to stick the different kinds of pollen grains on the stigma region. So if it is not compatible, naturally, stigma region never permit this pollen grain to sprout. Then scientists identified many such chemical to make the pistil to accept pollen for intraspecific hybridization. Intraspecific hybridization means what? The hybridization cross between what the individual of two different species. It is not possible normally to sprout the pollen grains on the stigma region of what another species. But after identifying such type of chemical or chemical nature of what the things that germinating fluid present on the stigma, we can apply it on the stigma region, so we can make some other pollen grains to sprout on the stigma. That is what the intraspecific hybridization, right? And this is what the pollen germination. Once it's identified, the stigma and the pollen grain matching each other, naturally that pollen grain starts sprouting, and it is called as what? The pollen germination. If it is mismatching, if it is not compactable, naturally what happens? It disintegrate on the surface of the stigma itself, never permitted to produce the pollen grains. Then come to the events in pollen pistil interaction. First, the recognition of pollen grain by the pistil only. That pistil identify this pollen grain belongs to the same species through what? This interaction only. Then if it is compactable, permit the pollen grain to prepare the pollen tube. Pollen tube start produce a pollen germination. And two-celled pollen grain divided into two male nuclei. We will see the details of events happening during this pollen, the pollen tube formation. There are two cells present in pollen grain, maybe remembering that vegetative cell and generative cell. And this Vatican cell divide and produce two male nuclei and make the pollen tube enter into the micropyle through filiform apparatus. There is what? A micropyle, there is small aperture in that the ovule, and through this aperture micropyle, with the help of the filiform apparatus, that pollen tube can enter into the ovule for this fertilization. So this is what the things happening after the pollen pistil interaction, right? That pollination, then pollen adhesion, attaching this pollen grain on the stigma region, then pollen hydration and pollen germination, then pollen tube enter into the stigma region, Then pollen tube grow through the style, and finally pollen tube enter into the ovule. Then after entering the pollen tube into the ovule, naturally that real process of fertilization take place in the plants. So this is what the formation of pollen tubes. See, many pollen grains are attached to the stigma region, but only one pollen grain is getting the opportunity to fertilize with the egg, because only one ovule present in one ovary. This ovary consists of only one ovule. So naturally it permit only one male gamete is getting to opportunity to fertilize. And it is producing what? A tube. Once it is identified as a compatible, naturally pollen tube will be produced through the style, and it will increase its length, and finally it's enter into this ovule through a small aperture. Here we call it as what? The micropyle, right? Then come to a small topic mentioned in your textbook, and we will see the details of events happening after this pollen tube formation later. And there is a small discussion on artificial hybridization that is also mentioned in your textbook. And it is purely artificial. Artificial means what? The man-made only. And hybridization, selecting two parents of different traits, and our aim is to combine these two traits in one individual, and that process only we call it as what? The hybridization, and product is called as what? The hybrid. And it is purely artificial. Select two parents of different character. We need to combine two desirable character in one individual. For that, we have to select two parents of different character, and select a flower from each plant of the same growth, right? That the same kinds of flower we have to select from both the parents, and one we have to select as what? The male parent, and another one is what? The female parent only. And this female flower, we have to go for what? The emasculation. Emasculation means what? Removal of all the male reproductive part. All the male reproductive part and there can be removed by using Vatican scissor even before getting the maturity. See, male reproductive part can be removed by using the scissor. And this process is called as what? The emasculation. And actually emasculation is for converting a bisexual flower into unisexual, that is female flower only, because we selected this flower as what? The female flower, right? Emasculate one flower to make it female. Emasculation means what? Remove all the male reproductive part before getting maturity. So naturally that flower will be converted into a female flower only, and bag the emasculated flower to avoid the pollination. So after emasculation, we have to cover that flower with a polythene bag to avoid unnecessary pollination with the unwanted pollen grains. Then artificial pollination. After the maturity of both the flowers, we have to collect the pollen grain from the selected parent, and this we have to stick to the stigma region of that selected female flower with the help of some soft brush, and again we have to cover the flower with the polythene bag. That is also true. Even after pollination, chances are there to pollinate the flower with some unwanted pollen grain. To avoid that chance, again we have to cover the flower with the polythene bag even after this artificial pollination. And this is what the process of emasculation. As I told you, remove all the male reproductive part. This is a bisexual flower only, having both male and female reproductive part, and the male reproductive part can be removed by using what? The scissors before getting maturity. So naturally that bisexual flower will be converted into what? A female flower only. Bisexual one will be converted into female flower. And this flower, immediately after emasculation, we have to cover it with the polythene bag to avoid the pollination of this flower with unnecessary pollen grain, and that only we call it as what? The bagging. And afterwards we can go for artificial pollination. Collect the pollen grain. After getting maturity, that pollen grain we have to collect with this brush, then that can be stick to the stigma region. So naturally after this pollination, again we have to cover the flower with the polythene bag to avoid unnecessary pollination again. So naturally they will go for that pollen pistil interaction, and finally they identify the pollen grain of the same species, then remaining that the pollen germination fertilization will take place later, right? Then come to the real process of fertilization happening in that angiosperm, and we call it as what? The double fertilization. We will see why it's called as double fertilization, and this is what the special type of fertilization happening in the angiosperms. See, after this pollen germination, this pollen tube grows towards the ovary. Ovary is located at the bottom portion only, and through the style only it moves from the stigma region by producing what? The pollen tubes. Here we have seen that pollen tube formation. Here we can see after the pollination, after the pollen interaction, it is permitted to grow through this what you can say style, and it will reach to the ovary. So this is what the process of what? The pollen tube formation. See, after pollen germination, pollen tubes grow towards the ovary through the style, and pollen tube brings both generative and vegetative nuclei towards the ovary. There are two nuclei present in the male reproductive gamete, pollen grain. One is called as what? The vegetative, and another one is what? The generative nuclei only. So through this pollen grain, these two generative and vegetative nuclei are taken to the ovary for fertilization. And just before reaching the pollen tube near to the micropyle, whenever it comes near to the micropyle, and this is what? See, this one is what? The micropyle. There is a small aperture, and whenever it comes close to the micropyle, and through the micropyle only, this ovule is taken inside. And before taking this pollen tube inside the ovule through this micropyle, what happened? This vegetative nuclei degenerate, and only what? The generative nuclei only, vegetative nuclei and degenerate and disappear. Vegetative nuclei only degenerate and disappear, and only generative nuclei remains there. Okay, there are two nuclei, want to enter into the ovule through the aperture, generative nuclei and vegetative nuclei. And vegetative nuclei, before entering into the ovule, get degenerated and it disappear, and only generative nuclei there. And this generative nuclei immediately go for a division mitotically. Mitotic division means what? The equational division only. See, after dividing this generative nuclei, and it produce what? The two haploid nuclei, and we call it as what? The sperm cell, or we call it as what? The male nuclei. So you know generative nuclei is haploid, already it is haploid. Whenever this generative nuclei undergo this process of mitosis, it produces two male nuclei, and both the male nuclei are also haploid, and this male nuclei only enter into the ovule through what? This micropyle. Micropyle is what? A small aperture. Through this aperture, these two cells called as what? The sperm cell or the male nuclei, both are haploid only, it's entering inside the embryo sac. Then pollen tubes enter into the ovule through the micropyle and take both male nuclei in the embryo sac through the filiform apparatus. Filiform apparatus means what? Here on either side we can find, here you can find the picture. Here you can find the synergy cells on either side, and egg is located exactly at the center. And these synergy cells are having some kinds of what? The cytoplasmic extension, and that only we call it as what? The filiform apparatus. And through this filiform apparatus only, pollen tubes are getting into the embryo sac. In embryo sac only, this egg cell is located, and polar nuclei both are located in the embryo sac only. The purpose of these two cells to fertilize with these two structures, and it is present in the embryo sac only. So it is entering into the embryo sac through such a filiform apparatus only, through the filiform apparatus. So pollen tubes enter into the ovule through the micropyle, and take both male nuclei in the embryo sac through that filiform apparatus present on the synergids, right? Then this is what? The pollen tube towards the ovary. Pollen tube is moving towards the ovary, and these are the two generative and vegetative cell. It is also taken to the ovary only for the fertilization, and it is moving and finally is reaching here in the micropyle end. Whenever it comes near to the micropyle end, that vegetative cell get degenerated and disappeared, and only the generative cell that go for this mitotic division and produce what two male nuclei, right? That only getting the opportunity to enter into this embryo sac for this double fertilization. So let us see what happened to this male nuclei after entering into the embryo sac with the help of this Philippium apparatus present in the synergids, right? And one male nuclei after the entry into the embryo sac and fused with what egg cell present in the embryo sac form a zygote called as what the syngamy. This is what the first event. But before the syngamy, sometime chances are there to fertilize the two polar nuclei present exactly at the center of this embryo sac. Remember, in embryo sac, there are two nuclei at the center, and both are haploid only. And even before syngamy, chances are there to fuse both polar nuclei together to form a diploid cell. And one male nuclei immediately after enter into the embryo sac, and this male nuclei or the sperm cell fused with the egg cell only. Egg cell present in the micropyle end. It's located in between the synergid cell and produce what a diploid cell, and this diploid cell produce due to the fusion of what this male nuclei and egg cell is called as what the zygote, and this process of fusion is referred as what the syngamy. Whenever this question comes in the examination, you have to mention two, the syngamy and the triple fusion, while discussing about what you can say that a double fertilization. So there are two events happening during this double fertilization. One is syngamy. It is the process of fusion of male nuclei or the sperm cell with the egg cell in the embryo sac only. Then other male nuclei, there are two male nuclei or the sperm cell. It's enter into that embryo sac and fused with the two polar nuclei present at the center. And both polar nuclei are haploid nature and form the PEN, that is primary endosperm nucellus. PEN, primary endosperm nucellus. And this is called as what? Triple fusion. Why it is called as triple fusion? Because here three haploid cells fused together. Right, two polar nuclei already present at the center of the embryo sac, fused with one male nuclei. So N plus N plus N. Three haploid cells fused together to form a triploid cell, and this triploid cell only we call it as what? Primary endosperm nucellus. Why it is called as primary endosperm nucellus? Because later stage this primary endosperm nucellus only will divide and produce what? The endosperm for the storage of food, right? That development we will see later. And whenever we are discussing about the double fertilization, we have to mention two things here. One is what? The syngamy, and another one is triple fusion. And this fertilization in angiosperm is not like what any other individual. Here there are two fertilization. There are two fertilization. One is a fusion of male nuclei with the egg cell. We call it as what? The syngamy. And another one is what? The fusion of another male nuclei with this this polar nuclei present exactly at the center of this embryo sac, and we call it as what? The triple fusion. There are two events here, syngamy and triple fusion. That is why we call it as what? The double fertilization. Two fertilization. One is zygote and another one is what? The triple fusion only. Once again, that syngamy is fertilization only. Here we can find the fusion of one male nuclei with the egg cell present in the embryo sac, and another one is what? The triple fusion. There we can find the fusion of three haploid cells, and two are there in the center called as what? Polar nuclei, and it fused with what? Another male nuclei and produce what? A triploid cell, and it is called as what? The triple fusion, right? This is what? The double fertilization. So release of sperm cells, the male nuclei into the embryo sac. And here one male nuclei fused with the egg cell present here, that only developed to become the zygote, and another male nuclei fertilized with the two polar nuclei exactly located at the center, and that only developed to become a triploid cell, and it's called as what? Primary endosperm nucellus. So there are two fertilization here, one fertilization fusion of the male nuclei with the egg cell, and another one with these two polar nuclei present here at the center of the embryo sac. And it is only, we call it as what? The triple fusion. And the product of triple fusion is what? The zygote. This is what the product formed here after the fertilization. And the product of what? The syngamy is called as what? Zygote. One male nuclei with the egg cell, syngamy only, is product is called as what? The zygote. And another product formed here due to the fusion of three cell, and we call it as what? The triple fusion only, and it is what? The primary endosperm nucellus, and that only developed to become endosperm in the later stage for the storage of nutrients. Then here, these are the sperm cells entering into what? The embryo sac through the filiform apparatus present in the synod. This is called as what? The yellow colored one is what? The synergids, and that pink colored one is what? The egg cell. There are three cells in the micropyle end of an ovule, and through this micropyle, this pollen tube enter inside and release this male gamete. That is what? The spore cell. By using that filiform apparatus, and one male nuclei fused with the egg cell here, and that is called as what? The syngamy, and produce zygote, and another male nuclei fertilized with the central cell, that is what? The polar nuclei. There are two polar nuclei here right the center, and it is called as what? The triple fusion, and collectively we call it as what? The double fertilization only. And see the double fertilization here, this is what? The egg cell, and these two are polar nuclei, and this is what? The filiform apparatus. You can see such type of outer cytoplasmic extension of synergids, that only helping the male gamete to enter into the embryo sac. Once this enter into the embryo sac, one male nuclei fuse with the egg cell here, another male nuclei fertilized with the two polar nuclei present at the center. And this is what? The product after this fertilization, and this is what? The zygote it produced here in the micropyle after fertilizing one male nuclei with the egg cell, and this is what? The central cell, or we call it as what? The primary endosperm nucellus, and it is what? The triploid cells only. And here we can see what you can say, antipodal cells. Then here we can see the synergid cell, and it is not having any kinds of significance, so naturally that get disintegrated, and these nutrients from these cells also will be utilized by primary endosperm nucellus cell as well as the zygote for the development. And through plasmodesmata, it can be reached to what? The primary endosperm nucellus, so it go for further development. Right, come to the other details of the triple fusion. As I told you, this is what? The process of fusion of one male nuclei with two polar nuclei, and it is called as what? The triple fusion. Two polar nuclei located at the center of the embryo sac, that only fertilized with this male nuclei and produce what? A single triploid cell, and it is called as what? The primary endosperm nucellus. Primary endosperm nucellus. This is what? The product of triple fusion. And primary endosperm nucellus further go for division and development, and this will be converted into endosperm, and it is exclusively for the storage of food material, and this food only utilized by the fertilized egg or the zygote to develop, become an embryo. Embryonic development from the zygote only. For the embryonic development from the zygote required nutrient that will be provided by the endosperm only. And this food is used by embryo for its development from a zygote only. And this process takes place before the development of embryo. right? Actually, there are two type of development happening after fertilization in the embryo sac. One is the product of syngamy, that is zygote only developed to become an embryo. And second one is what the development of primary endosperm into endosperm only. And the primary endosperm will be converted into endosperm only for the storage of food material, right? And for this food material stored in the endosperm used only for the development of zygote into embryo. So naturally, after double fertilization, primary endosperm only go for a division and development, and it will be converted into endosperm and store food material. And after the storage of food material in the endosperm, that food only utilized by the fertilized egg to convert into an embryo. So this process takes place before the development of embryo from the zygote. So for the development of embryo from the zygote required nutrient, that will be provided by the endosperm only. So first, immediately after double fertilization, that the product of the triple fusion, primary endosperm nucleus undergo the development and produce endosperm and store food material. And after the storage of food material, this food only utilized by the zygote to develop to become an embryo. That always remember that. Then come to the development of endosperm. How endosperms are getting developed from primary endosperm nucleus. See, primary endosperm nucleus is what the product of triple fusion only. Whenever a male nuclei fuse with the two polar nuclei, there will be a triple cell at the center of the embryo sac. That only we call it as what the primary endosperm nucleus. And this primary endosperm nucleus only undergo further development to form this endosperm. That only we are going to discuss here in very detail, right? Then endosperm developed before the embryonic development. Why should go for development before the embryonic development? Because for the embryonic development, nutrients are very much required. And this nutrient actually provided by the endosperm only. So endosperm should develop before the development of this embryo from the zygote, right? Then primary endosperm divide repeatedly. This primary endosperm, it is a single cell only, a triploid cell only. It's a product of triple fusion that divide repeatedly through mitosis and produce a mass of cell in the embryo sac. That only we call it as what the endosperm. At the center of the embryo sac, a mass of cell will be produced by this PEN, primary endosperm nucleus, through the repeated equational mitotic division. And this is made with the triploid cells only. All the cells of what the endosperm is made with the triploid cells only, and it store a lot of food material inside. And the food from PEN, primary endosperm nucleus, only used by the zygote for the development into embryo. So the food material stored in the endosperm, PEN, will be converted into endosperm and store a lot of food material. All the cells of the endosperm is what you can say triploid cells only. That food stored in the endosperm only utilized by the zygote for its development into embryo, right? This is what the development of the PEM, that is primary endosperm nucleus, into what this endosperm, and it is formed here, and finally it go for division repeatedly, and division is what the mitotic division only, and produce what a mass of cells inside the embryo sac. And this mass of cell only store the food material. All the cells are triploid because these are all developed from a primary endosperm nucleus. Basically is a triploid cell through the process of mitosis only. Then come to the development. There are three ways of development of endosperm for the storage of food material. One is what the nuclear, second one is cellular, and third one is what the combination of both, we call it as what the halobial type, right? Nuclear type, cellular type, and the halobial type of development is common in endosperm. Endosperm development mainly in three ways. The nuclear development, simple concepts only. In case of nuclear development, the cells of the primary endosperm divide. Primary endosperm nucleus divide but only its nucleus repeatedly without the cytoplasmic division. And division is mitotic equational division only, but only nucleus will divide. Or single cytoplasm, we can find many nucleus. And later what happened, every nucleus will converted into what the cells repeatedly due to the cytoplasmic division, and such a type of development is called as what the nuclear development of endosperm. Initially, the nucleus only will undergo the process of division repeatedly through the mitosis. And another one is cellular. Cellular means what? Nucleus will divide followed by the cytoplasmic division. Repeatedly, the cell will divide and produce a mass of cell in the embryo sac, and that only acting as what this endosperm for the storage of food material. And another one is what? The halobial. It is what? The combination of both the nuclear and cellular only. In halobial, first it divide its nucleus for a period of time. For a period of time, the nucleus will divide repeatedly. Then it's followed by the cytoplasmic division and cellular division, right? That after completing the nuclear division, go for cytoplasmic division and produce mass of cell, and this mass of cell again undergo the process of what? Cellular division for many times and produce a mass of cell at the center of the embryo sac. It is actually the combination of both the nuclear and cellular type of development only, and referred as what? The halobial type of endosperm development. Endosperm is actually developed from the product of triple fusion located exactly at the center of the embryo sac. It is triploid that divide mitotically and produce a mass of cell. And this cell is triploid cells only able to store a lot of nutrient, and this nutrient only supplied to the zygote for its development. Right. This is what division primary endosperm nucleus exactly form at the center of this embryo sac. It divide repeatedly and produce what? The many cells inside. Right here, it is cellular. It go for division, and there are three ways to divide this cell. One is what? The nuclear. Only nucleus will divide. Later, the cytoplasm will divide evenly. Then cellular, all the cell will divide. Nuclear division followed by cytoplasm. And halobial, it is what? The combination of both the nuclear and cellular development, right? And produce what? A mass of cell inside, right? And it is what? The endosperm. It is fully filled in the space of the embryo sac, and this cells only store the food material. And during this development, embryo will be there. I mean that the zygote will be there in the micropylon. And this start development only after completing the development of endosperm and store the food material inside, because the development of zygote into embryo required a lot of nutrient that will be provided only by the endosperms. Then this is what the endosperm development, and this is what the development of the embryo in the micropyle end only, right? Then come to the development of zygote. You know, zygote is what? The product of the zygami, male nuclei. After enter into the embryo sac, it fertilized with or fused with the egg cell present in the micropylon and produce a zygote. And this zygote only developed to become what? An embryo later stage through the cellular division. You remember, it is not triple fusion. It is what? A fusion of two cell only. One is egg cell, it is haploid, and another one is male nucleus, that is also haploid. So naturally, zygote is diploid, and this diploid zygote only divide and go for development and produce what? The embryo. So naturally, embryo also will get the diploid number of chromosomes. And this embryo only later stage developed to become what? A plantlet. Embryo only developed to become a plantlet later. See, zygote start divide along with the endosperm development. See, after the development of the endosperm and storage of food, the zygote also start dividing, and zygote divide into two. Initially, it divide into two mitotically and produce two uneven cells. Two uneven cells. Here you can see two uneven cells. This is what the zygote, and this zygote is diploid, and this diploid cell divide and produce two uneven cell after the first division. and one is towards the micropyle, that is what the larger one, we call it as what the basal cell, and another one is directed towards the chalasalin, and we call it as what the epibasal cell. Basal cell and epibasal cell, these are the two cells produced from the zygote after the first division, and this division is what simple mitotic division only. And later stage, that basal cell will never go for the further division, whereas epibasal cell divide and get what the structure like this, right? It go for continuous division and development and get what a heart-shaped structure in embryo, that again will go for further development to produce what the plumule and radicle, and also getting what the two cotyledon in case of dicot plants. And this is what the structure they called as what the cotyledon for the storage of food material. That is what the structure in the embryo. There are two structures you can study here for the storage of food material. One is what the endosperm. The food stored in the endosperm is completely utilized during the development of what the zygote into embryo. It is for the embryonic development only, whereas the food stored in the cotyledon is used for the development of a plantlet from the embryo. You know, this is what the embryo, and this embryo only developed to become a plantlet. You know, embryo consists of what? Embryonal axis. And there are two regions, one is plumule and another one is radicle. And plumule is actually the future shoot system, and radicle is what the root system, future root system. And root and shoot system of the plantlet is developed from the embryonal axis only by providing food material stored in the cotyledon. So the food stored in the cotyledon is completely utilized for the development of the embryo into plantlet, whereas endosperm is also storing food material, but this food is utilized only for the development of what? The zygote into embryo. Zygote into embryo, then embryo into a plantlet only. So that only we are going to discuss here in detail, development of zygote into embryo. See, zygote start dividing along with the endosperm development, and first zygote divided into two, and mitotic division only, and produce two uneven cells. The large cell, as I told you, is directed towards the micropyle, and it is called as what? The hypobasal cell or the basal cell, and small cell directed to the chalasalin, and that is called as what? The terminal cell or the epibasal cells only. This is what the product after the first division, uneven division only, but mitotic division. Two cells, one is larger, and we call this larger cell as what? The basal cell, that is projected towards the micropyle end inside the embryo sac only. And another one, it is what? The epibasal cell, and it is directed towards what? The chalasal end, and it is very small in size. And this epibasal cell only undergo further division to produce what? The structure like this called as what? The suspensor, and also helping for producing what? A heart-shaped embryo. And this cell again will divide cells of this heart-shaped embryo again for division and development, and produce what? Two structure here for the storage of food, and we call it as what? The cotyledon. And here you can find an embryonal axis consists of two poles. One is what? The radicle, and another one is plumule. This is what? The matured embryo consists of what? The radicle, plumule, and also containing a structure called as what? The cotyledon, and it is exclusively for the storage of food, right? And see the development inside. This is what? The first division, and produce what? The uneven cells only. And again what? It go for producing this heart-shaped structure very slowly, producing the heart-shaped structure here. Here you can find what? This structure, and here the heart-shaped structure. And there is an axis here. There is an axis here. One is what? The radicle, and another one is what? The plumule only. And this structure is collectively called as what? The embryonal axis. This area is called as what? The embryonal axis, with the two cotyledon. Two cotyledon. This is what? The cotyledon. These are the cotyledon, and it is what? The what you can say, embryonal axis, and this one is what? The suspensor. Actually, the purpose of the suspensor is to push this embryo towards the food material. As I told you, it is during the embryonic development, it consumes the nutrient present or stored in the endosperm only, and suspensor are only pushing this embryo toward the endosperms to receive the food material for its development. And finally, it produces, this is what the ovule, this is what the ovule, inside the ovule, embryo is getting developed, and these are the cotyledon, and this is what the embryonal axis consists of both the plumule and radicle. Here plumule, here you can see the radicle, and this is what the suspensor, and this one is what the basal cell. Basal cell always directed towards the micropyle only. This is what the micropyle, you can see the micropyle here, and towards the microspile only it is directed, this basal cell, and it is what the suspensor only pushing this embryo towards the direction of the endosperm to receive the food material stored in the endosperm. And there are two structures also developed in the embryo during its development. One is what this embryonal axis. This embryonal axis only developed to become a planlet later. It consists of both the plumule and radicle, and another one is what this cotyledon, and cotyledon is like endosperm. It is exclusively for the storage of food. So another different, the endosperm, it is triploid cell. Endosperm is for the storage of food, and food is used for actually the development of what this zygote into embryo. And it is triploid cells only, made of a number of triploid cell, whereas cotyledon, it is made with a diploid cell only, and that also used for the storage of food material, but the food stored in the cotyledon for the development of this embryo into planlets only. Right. Then come to the embryogenesis. That is what the formation of embryogenesis, formation of embryo. These are all what the events happening during the development of embryo. And the process of development of the embryo from the zygote is called as what the embryogenesis. As I told you, it's divided unevenly. First division, second division, third division, fourth division, like it divide, keep on dividing and produce what the pro embryo like this, and finally it go for a type of rearrangement, getting what the shape like this with the two cotyledon, then suspensor, and this is what the basal cells. Right, the same thing, zygote divide initially into two and produce what two uneven cell, and here it go for the next division. This cell only will divide later and produce what the suspensor, many cells one after another. It's arranged with what a round globe shaped embryo. We call it as what the globular embryo. And this globular embryo, the cells will divide repeatedly and arrange in a specific way to get what a heart-shaped embryo. And this heart-shaped embryo again go for the division of this diploid cell and produce what embryonal axis here consist of radicle and plumule, and these are the two cotyledon. Embryonal axis, this end is what the radicle, and here it is what the plumule. And radicle only later developed to become the root system of the future plant, and plumule developed to become the shoot system of the future plant. And for the development of radicle into root and plumule into shoot system, they are using the food material stored in the cotyledon. So this is what the type of development in after this double fertilization, after development of zygote into embryo, after double fertilization, and we call it as what the embryogenesis. So embryogenesis is nothing but the process of formation of embryo from the fertilized egg, or we call it as what the zygote only, and this is also happening inside the embryo sac. Right. Then come to the special mode of reproduction. This is what the fertilization events. Now we are discussing about the different kinds of event happening during the fertilization in plant. We have completed the pre-fertilization event. Pre-fertilization event includes formation of male gamete and female gamete, gametogenesis. Then we have seen the process of pollination and pollen tube formation. These are the three events happening before fertilization. Then during the fertilization, we have seen that two type of fertilization, collectively called as what? Fertilization event. And after fertilization, that is what the post-fertilization event, that product of the triple fusion will be converted into endosperm, and the product of syngamy, that is called as what? Zygote, that developed to become what? The embryo. That is called as what? The embryogenesis. Then come to what? The special mode of reproduction. Right. One is what? The agamospermy or apomixis. And here the seeds are formed without the fusion of gametes. So what will happen to this ovule after fertilization? After fertilization in the ovule, there are two structures. One is what? Endosperm, and another one is embryo. See, some food material present in the endosperm will be utilized by the embryo during the development, and fully developed this embryo, not having what? The endosperm inside. So later, this ovule only developed to become the seed. And sometime we can see the formation of seed even without this double fertilization or fertilization of the gamete. And it is called as what? The apomicte. Product is called as what? The apomicte. Apomicte is nothing but the seed produced because of what you can say, some reasons mentioned here, we will see it later, and some reasons helping this ovule to develop to become the seed even without the fertilization. And this product, that seed produced from the ovule without fertilization, is called as what? The apomicte. And process of formation of this seed without the process of fertilization is called as what? The apomixis or agamospermy. Then let us see what are the reason for the formation of what? Such type of seed without fertilization, or what are the reason for the apomixis or agamospermy. Then first one, nucellus become an embryo. You may remember that structure of the ovule. In that ovule, embryo sac is there, and this embryo sac actually developed from the nucellus cells only. But many nucellus cells are there outside this embryo sac. Sometimes these nucellus cells can develop to become an embryo because it is already diploid. You know that diploid zygote only normally develop to become an embryo, because some reason nucellus cell can also develop to become an embryo because that is also diploid cell. So their embryo formation will be there without fertilization. And synergy cells are there in the micropyle end, and this on either side of this egg cell. Instead of fertilizing the egg cell with the male nuclei, two synergy cell can go for fusion. Both the synergy cells are haploid in nature. Whenever the synergy cell go for fusion, it produce what? A diploid cell, and sometimes this diploid zygote can develop to become what? A planlet. Planlet means what? An embryo. That is what another reason for the apomixis. Or sometime egg will be produced without meiosis. That means egg is having what? Diploid number of chromosomes. Normally, that egg is haploid because of what? The meiotic division happening during the development. And sometimes what? Egg will be produced without this meiosis. So naturally, that will be diploid. And such a type of diploid egg can develop to become an embryo. And that is what another reason for the formation of seed without fertilization, and we call it as what? The apomixis, and this product, the seed, is called as what? The apomicte. Then come to another term. Such type of terms you have to by heart as it is, because chances are there for asking such type of question for multiple choice, right? And parthenocarpy is what? Another term. And here it is like apomixis only, but in apomixis, formation of seed only without fertilization. But here it is what? The development of fruit from unfertilized flowers. Actually fruit is developed from the ovary. After fertilization, ovary developed to become the fruit, and ovule present inside the ovary developed to become the seeds. And some cases, chances are there to develop these fruits from unfertilized flowers, and it is called as what? The parthenogenesis. And normally it produces what? The seedless fruits. There is no seed because no fertilization. So after fertilization only, that ovule will be converted into seed, and here no fertilization, so no chance of any kinds of seed formation, but this ovary wall can be thickened and it can be converted into fruit formation without fertilization and no seed inside. And like a pineapple, some grapes you may be knowing, seedless grapes, these are all produced fruits without this fertilization only, and that is really referred as what the parthenocarpy, and this can be produced by using some kinds of hormone, particularly in tomato. Now that seedless tomatoes are available, then seedless grapes are available, we are making such type of what the fruit commercially without seed inside, artificially through what you can say same process, that is parthenogenesis only. Development of seed from unfertilized egg, and it may be diploid or it may parthenogenesis, it may be development of seed from unfertilized egg only, it may be diploid or haploid, depend on the nature of the egg. Sometime I told you that egg is what diploid. If it is diploid, seeds will also develop from this unfertilized egg. So naturally seeds, it is diploid in nature, and sometime what it's developed from the haploid cells only. So naturally that will be haploid in nature, right? Then come to next term, that is what the polyembryony. Polyembryony means normally one what you can say ovule present in one embryo. More than one embryo in an ovule. Normally one ovule consists of one embryo only, right? One embryo in one ovule. But sometimes we can find more than one embryo in an ovule. More than one embryo. Every embryo can become a new plantlet. That means one seed produces what many plantlets. Ovule only develop to become the seed later, and this seed can produce many plantlets in case of what the polyembryony. So polyembryony is presence of more than one embryo in one ovule, and it may be due to more than one embryo sac in one ovule. So normally one ovule consists of only one embryo sac developed from the nucleus cell only. Sometimes chances are there for many embryo sac in one ovule, because it is developed from many numbers of the nucleus cell, and it may be due to the development of more embryo from synergids, antipodal cells, and nucleus only. Development of more embryo, chances are there for the fusion of the synergid, that only can act as what the zygote, or sometimes antipodal cell can go for fusion, can also act as what the zygote, and sometimes nucleus cells already it is diploid, that can also act as what a zygote, and chances are there to produce many embryo inside the same ovule. So naturally that seed developed from the ovule can produce many number of plantlets, because every embryo developed to become what a new plantlets only, right? Then come to the post-fertilization. That means events happening after the fertilization, post-fertilization events, right? See, after fertilization, see the condition of the ovary, and the ovary wall developed to become a fruits, and many ovules will be there in this ovary. That ovary after fertilization developed to become the seeds, and every ovule there is a protective structure called as what the integument, and this integument only developed to become the hard coating for the protection of the seed. We call it as what the seed coat. And embryo sac, this is what the structure present in the ovule, that only developed to become that seed membrane. There is a membrane in the seed just below what the seed coat. And PEN, primary endosperm nucleus, that we already discussed, it is what the product of triple fusion that developed to become the endosperm. And fertilized egg, it's a product of syngamy that developed to become what an embryo, and later embryo developed to become a plantlet. So these are what the events happening after the fertilization. We call it as what the post-fertilization events. Once again, that ovary wall, see, wall of the ovary get thickened and become more fleshy, and that only develop to become the fruits, and many ovules will be there inside the ovary, and every ovule present in the ovary after the fertilization develop to become the seed. And every ovule is having what one or two layered protective structure called as what integument, and that also cover the seed, and it is very hard and impermeable even to the water, and we call it as what the seed coat. And inside the ovule, embryo sac is there, and this embryo sac only developed to become the seed membrane. Then, as I told you, the product of what the triple fusion PEN, primary endosperm nucellus, developed to become endosperm, and the product of syngamy is a fertilized egg that only developed to become the embryo, and later embryo developed to become the plantlet after consuming the food stored in the cotyledon, right? And this is what the female reproductive part. What will happen to this after this double fertilization? So all these things are not having any significance after fertilization, the stigma and style. So that area get disintegrated, and this area ovary is attached to the plant permanently there, right? And this ovary, after a period of development, that ovary get thickened, become more fleshy, and that only develop to become the fruits. And many ovules will be there normally in one ovary. And after double fertilization, every ovule develop to become what the seed only. And this is what the integument, the protective structure of the ovule by having a micropyle here. And this integument is a covering of this ovule developed to become what the seed coat. It is for the protection of seed, and it became very impermeable, and gas exchange and exchange of moisture, everything happening through the micropyle only. Even after the development of ovule into seed, we can find the micropyle, and embryo is there inside, and it is living only, and it required what you can say water and oxygen. Gas exchange should be there. So all these things are getting exchanged through the micropyle, small opening present in the seed only. And integument developed to become the seed coat, and this embryo sac, you can find this embryo sac here, and this embryo sac only developed to become this membrane inside the what you can say that seed coat, right? So this is what the fate of different parts after fertilization. And here the primary endosperm, and it is what the product of triple fusion, as I told you. And this primary endosperm developed to become the endosperm, and this food stored in the endosperm will be utilized by the zygote to develop to become what? An embryo. And later embryo is having plumule radicle, and this plumule radicle developed to become its shoot system as well as the root system by consuming food material in the cotyledon, right? And see that development of fruit from the ovary. This is what the ovary. This is the ovary part of the flower, and this ovary only developed to become the fruit in the late stage, this portion. And many ovules are there in the ovary, and these ovules only develop to become what? Seed inside. So this is what the development of what? Fruit from this ovary. And come to the next one, the structure of a fruit. And fruit is having a tough covering that is called as what? The epicarp. And there is a thick region, and it is very fleshy and sometimes very sweet in case of mango, and it is called as what? Mesocarp. And there is a tough layer inside the mesocarp that is called as what? The endocarp. And inside the endocarp only seed is located, and seed consists of what you can say three things, as I told you. That embryo is there, that embryonal axis, then cotyledon. And embryonal axis consists of what? The plumule and cotyledon that is there in the seed that only develop to become the plantlets, right? This is what the structure of the fruits, and it is what the pericarp, outermost one, exocarp, mesocarp, and endocarp. And this is what the placenta, and placenta only provide the nutrients to the ovule for the development, and this ovule present in the locule, these are all what red colored one you can see here, and it is called as what? The compartment or the locule. Inside the locules only every ovum after fertilization develop to become the seeds, right? Then it is what? Another lemon. Same thing you can see with what you can say mesocarp, endocarp, then exocarp here, outermost layer. It is what? The middle layer mesocarp, and it is what? The inner endocarp. And these are the locules only. Locules where we can find the ovule, and after fertilization, every ovule does become what? The seeds only, right? Then it is what? The fruit. You can find this epicarp, outermost tough one, and mesocarp is here, and it is not very fleshy. It is fibrous in case of coconut. And here you can find this what you can say endocarp, right? This is what the innermost layer, we call it as what? The endocarp only. Exocarp, mesocarp, and this one is what? The endocarp. And the innermost one, this is what? The testa layer, right? That brown colored thing, this is what? The testa layer, right? Then come to another concept, that is what? The false fruits. Now we have seen in detail about the structure of a fruit. Then let us see that false fruits. What is false fruit? It is not real one. So real fruit, normally it is developed from the ovary only after fertilization, whereas the false fruits can develop from any other part of the flower, right? Example, you can find apple, strawberry, cashew nuts. Here, the fruits are developed not from what you can say ovary, it is from some other parts only. And particularly that, the cashew nuts, that the fruit like a structure, a fleshy one, a sweet one, develop from the thalamus only. And such type of fruits only we call it as what? The false fruits, not the real fruit. Real fruit always develop from the ovary only. Ovary wall get thickened and become fleshy, and that only develop to become the fruits in case of what? The real fruit. And true fruit, and it is called as what? The false one. Examples: apple, strawberry, cashew nuts, etc. And fruits normally develop from the part other than what you can say ovary of a flower is called as what? The false fruits, right? Apple, then strawberry, cashew nuts, these are all example for the false fruit only, because these fruits are developed from the thalamus region. This is what? The false fruit. See, actual fruit is what? This is what? The ovary portion of this fruit, and this is what? The fleshy one. This one is what? Very fleshy and sweet in taste, and it is developed from the thalamus region only. That is why we call it as what? The false fruit. Similarly, strawberry also is fleshy. That sweet one is developed from the thalamus region only. And here in case of what? The apple, this is what? The real part of the ovary, and this is what? The thalamus, that only become fleshy and thick, and develop to become the fruits. That is why these apple, strawberry, and cashew, these fruits are not called as what? The real fruit. They are false fruits only, because these fruits, fleshy thing, a sweet one, is developed from the thalamus region of these flowers only. Then come in detail about the structure of seeds. So all of you know the seeds are developed from the ovules only. After fertilization, ovule only develop to become the seed. Inside the seed, mainly we can find embryo, and embryo consists of what? This the three things: embryonal axis, plumule, and radicle, and also containing what? The cotyledon for the storage of food. So seeds are developed from the ovules. Integument of ovule, already we discussed, developed to become the seed cord for the protection. And embryonic sac developed to become the seed membrane. Embryonic sac present inside the ovule, that only developed become the membrane just below the seed cord. And primary endosperm nucleus is utilized for it is converted into endosperm and full totally utilized during the development of the embryo from the zygote, and later by using this food stored in the endosperm, zygote developed to become an embryo. And embryo consists of mainly two things. One is embryonal axis. Embryonal axis, there are two parts. One is what? The plumule, and another one is radicle. Other than this, cotyledon is also there. It is also storing food material. And this is what? The outer coating of a seed. It is called as what? The seed cord. and seed cord is developed from the integument only. There we can find a small opening. Maybe remembering the structure of the ovule, ovule is covered with bilayered this integument by leaving a small aperture, a small space that is called as what the micropyle. Even after the development of seed, micropyle will be there because you know that seed coat is impermeable to even water. So water cannot enter inside through this seed coat. So this exchange of gases and this entry of water inside the seed for the development only through the micropyle. Micropyle is what? A small aperture for the exchange of gases, right? And inside the seed coat, we can find the seed membrane. And this seed membrane is actually the embryo sac only, that, you know, membranous covering of the embryo sac only developed to become what this, what you can say, seed membrane. And inside we can find what you can say, embryo with the plumule and radicle here. It is what the radicle that only does to become the root system of the future plant, and plumule, it is what the future shoot system of the plant only. And the hilum, it is what a small marking in the seed where that attached to what the embryo, I mean, it is attached to the ovary wall in the placenta only. That is, that region is called as what the hilum, and it is what the cotyledon. Cotyledon is actually the part of the embryo only, but it is exclusively for the storage of food material. And this food stored in the cotyledon is utilized by this plumule and radicle to develop to become the shoot system as well as what the root system. And in case of dicot plant, we can find there are two cotyledon, whereas in case of monocot, only a single cotyledon. And because of that only, it's called as what? Monocot, monocotyledonous plant. If it is having two cotyledon, we call it as what? The dicot plant or dicotyledonous plants only, right? It is what? The dicot seed. See, this is what the seed of Bengal gram, having a brown colored testa coating. That is what the seed coat. Just remove that seed coat, you will get a membranous structure inside, that is called as what? The seed membrane. And here you can find the hilum. Hilum is what? A small marking of funiculus only, and this area only, this area of this ovule only connected to the placenta during its development, and supported with all kinds of nutrients. And the micropyle is here. It is a small opening, and through this opening only exchange of every material take place between the inner and outside of this seed. This is what the structure of the digote seed, and it is what the embryo. In embryo, we can find two cotyledon in case of dicot plant, and there is embryonal axis at the center, and this embryo is getting the nutrient from both cotyledon, and one part of the embryonal axis was the plumule that only developed to become the shoot system of the future plant, and another one is radicle that only developed become the root system of the plant, right? And it is what the monocot seed, and diagram should be by hearted. This monocot seed, right? This is what the endosperm, and it is scutellum, then coleoptile, the covering of the plumule. A membranous covering of the plumule in monocot seed is called as what? The coleoptile. And similarly, another membranous covering is there for the radicle also, that is called as what? The coleorhiza, and it is what? The embryonal axis. And this only developed to become a new plant later, and storing lot of food material here. And the seed is covered with what you can say, cover. We call it as what? The seed coat. For example, in case of rice, you can find a brown colored seed coat, and inside there is a layer called as what? Areola layer. In case of rice, you maybe find a brown colored a thin layer just inside what? The seed coat, and we call it as what? The areolar layer. It's full of vitamins, and storing lot of food material in the endosperm, that only utilized by even scutellum is there in monocot plant. It is nothing but the cotyledon only, and it is also stored for the food material, and food material stored in the seed only utilized by this embryo to develop a plantlet. This is what the structure of the monocot seeds. Then come to the viability of the seed. Viability. For every seed, there is a period of development that only we call it as what the viability. After that viability, it never sprout to become a plantlet. So seed going for germination and develop to become the plantlet. And these are the cotyledon. You can find two structure here. And this structure is called as what cotyledon. During the development of seed into plantlet, it utilize what the food material stored in these two cotyledon only, right? And after a period of its storage, it never go for germination because there is a period of viability for every seed. Afterwards, it never go for a sprouting, right? It varies from plants to plants. For example, in case of Lupinus arcticus of Arctic tundra region, and its viability is what 10,000 years. 10,000 years seed can be viable. It go for what you can say sprouting and produce a new flower, new plant. The 10,000 year is what the viability. Whereas in case of date palm, and it is viability is what 2,000 years. Whereas in case of our that Oryctes sativum, that ordinary paddy, and it's only for 1 year. After 1 year, it never go for a sprouting. That is what the period of viability of the seed. It varies from species to species, right? Then come to the inactive stage of the seed. See, after the formation of seed, you know, after the formation of seed, sometimes the seed will go for a period of inactive life. Actually, the seed sprout only when it get what a suitable climatic, a favorable climatic condition. Otherwise, it will be in an inactive stage only, and that only we call it as what the seed dormancy or quiescence. And these two concepts we will discuss after break. And next class will start from 6:30 to 8 o'clock. So we will see with this topic that seed dormancy and quiescence. These are the inactive stage of seed after development. It's waiting for a favorable conditions to sprout. Okay, thank you so much.